Our next speaker is Dr. John Hawkes, who's chair of the Department of Anthropology, and I'm going to mess this one up, I'm sure, Vilas Borghese Neighborhood, Distinguished Achievement Professor in Anthropology at the University of Wisconsin-Madison. And he has spent probably more time in Africa than I have spent outside the country. But going to talk with us about some of his findings and observations about early humans. I never get that word right, so I'm going to let you say what that is.
Sure.
He's going to take us way back with this and see where we go to get here. John.
Thank you so much. Thank you. Thank you, Tom. Last Friday, I was in Africa looking at new fossils, and I have to tell you that I am, as pleased as I am to be here. On WhatsApp, my team is giving me photos of the new fossils every day, and so I'm like, I'm missing it again. I will tell you that I was there for most of the last 6 weeks, and we added about 20 new human relatives to our docket of fossils that we work with. And so let me introduce you to my site. This is the Rising Star site. It's in South Africa, just outside of Johannesburg. It is pretty accessible. You could drive there. And this time of year, it's not this green. It's their winter time, so it's a little bit cold. There was frost in the mornings, and also a little bit browner than this. But it's a beautiful landscape, and I'm privileged to work there with some amazing people. I work in caves underground. I also work in laboratories. I work with an amazing team of people, and I want to introduce some of them. This is Professor Lee Berger, who is a National Geographic Explorer in Residence and South African and leader of our project. This is Dr. Kenelo Molipiane, who is, again, National Geographic affiliate and underground lead and lead excavator at one of the sites where I work, Gladysvale. This is Dr. Tebogo Makubela, who is at the University of Johannesburg and is our lead geologist. Everything that I do is a team enterprise. And I have, in the last 10 years, co-authored papers, scientific papers, with more than 250 people. And I like to say that at the beginning of every talk that I do, just to let you know that there's nothing that I do that is, adding meaningfully to our record that is not done with a very large group of people. And the collaborators that I have, some of them that I work with and am great friends with and see every time I go anywhere, and some that I might meet and work with on a particular project, and if that sort of thing comes up again, I'll probably work with them again. That collaboration is fundamental to all of the scientific progress that we see happening in our field today. Everything that we do is a huge group enterprise. Here I am in the lab last month with my graduate students. These are PhD students at the University of Wisconsin, and they're part of the work that we do. And training students, the next generation, is an incredibly important part of the work that we do. And I'm, of course, very committed to that. It's part of the reason why I'm here talking with all you, to make sure that everyone knows that this is a field that has exciting new things happening. There's new discoveries all the time. Young people are part of those discoveries, and we're looking for the best talent to be a part of them. The Rising Star Cave System is more than 3 kilometers of mapped passageways underground. So under the beautiful landscape that I showed you, there's a cave system that consists of 3 kilometers of underground tunnels. We work in varied parts of this system. And the part that I'm going to talk about first is the Dinaledi subsystem of this. The Dinaledi subsystem is where our first discoveries in the cave were made. These two guys, Rick Hunter and Stephen Tucker, in 2013, were helping us explore caves in the region for fossils. They were keen, they were young, they liked to go into caves, and they had this kind of, you might call it crazy attitude that some young people have that are interested in extreme caving, that they wanted to go into places where nobody had ever been. This is Stephen, and you're going to see some footage from his helmet cam as he's going down into this very narrow passageway. We named this the chute after we got working here a little bit. You'll see that the width of this is approximately 7.5 inches. And in order to go into this part of the system, you have to be able to fit there. I don't know if you can tell, but that's not me. But you see that he discovered on the floor of this chamber bones. And the photographs of those bones when they brought them out of the system, here you see a jawbone. And when we saw photos of this, we saw right away that this is a human relative of some kind. The 3 teeth that are on the right side of this, the left side of this, are the 3rd molar, the 2nd molar, and the 1st molar. And you see the 3rd molar is the biggest there. That's a trait that humans today don't have. How many of you all have your wisdom teeth still? A few of you. Most of you have them pulled. They're often very irregular in shape in humans. They're usually the smallest of the molars. And humans today have a big reduction of our teeth compared to many of our fossil relatives. This jaw was very much like some of our early human relatives, species that we call Australopithecus. And so we said, this is something very interesting. There was a skull outlined on the floor of the cave. And this demanded really rapid investigation. The challenge of this is that the cave system is quite challenging to move through. This was a different kind of cave than many of us had worked in before. Many of us work in much more open caves that have hard rock deposits of fossils, where the fossils are embedded in a rock, and they get that way because of the dripping of calcium carbonate with the cave dripping. And so that makes them a cement-like hardness. In this cave, we were looking at something that was much deeper, much less open, and something that we hadn't seen before. The contours of the system that we worked in to go to the Dinaledi subsystem are very challenging. There were multiple squeezes, and I can tell you that I go through some of these squeezes. How many of you all have been in a cave? Most of you, yeah. How many of you have been in a cave and they turn off the lights and they say, this is as dark as it will ever be? Yeah, they always say this, right? How many of you have been in a cave where the rock is pushing on you front and back in order for you to go through? Yeah, a few of you have been in that situation. That's what's called a squeeze, right? And those are not psychologically trivial. And to be honest with you, right, everybody has their own challenges. There is a squeeze to challenge everybody. And there are many squeezes in this cave that none of our team can manage. There are some that I cannot manage. The one that I'm going to talk about is one of them. And there are, again, some that are challenging for people that aren't me. And so we have a system where we make sure that everybody that is in our cave that's working with us is comfortable with the spaces that they're going to be in. People that go into some of the more dangerous parts of the cave are people who have quite extreme climbing training. And so we're working with a team of experts in the different elements. Some of the folks that work with me today underground are some of the most experienced people in underground work in the world, and certainly in the Southern Hemisphere. Folks that have spent more time underground than anybody. We got a team of very skilled excavators who could do this, who were climbers, who could fit through the squeeze, and began work in 2013. And the work that they did, you see an example of it here. In that time, the first month of excavation work, we recovered more than 1,200 pieces of bone. Those 1,200 pieces of bone, as they came out, we quickly perceived that they were not what we expected when we started. When we started, we thought, we're going to find parts of a skeleton. That's sort of what we often find. And very quickly, we had 3 upper right thigh bones, 3 different individuals. And as time progressed, we had more than 15 individuals represented in our site. The bones today represent more than 30 individuals throughout the cave system. And those 30 individuals have a fascinating combination of characteristics that we had not seen before in other relatives of ours that we'd unearthed. When we published our first description of these bones in 2015, it was an important event for us. It's not every day your photo's on the front page of the New York Times. And that was, one of a very few, it was the second biggest science story of 2015. And I hate to complain. There were these guys that built this spaceship that went to Pluto. And they thought that that was a bigger story than ours. It took a long time to get to Pluto, I will admit. We named this species Homo naledi. I had the name there for you, Homo naledi. Naledi means star in the language that's spoken quite commonly around this area of South Africa, Sesotho. Homo naledi has very many human-like characteristics. It has human-like hands, complete with thumbs that are proportioned relative to the fingers like ours are, broad fingertips and thumb tips that are made for applying force across that pinch grip, that joint, which we use for tool manufacture, a wrist that's shaped like ours with the bones configured like ours are. So it's very human-like in some respects, but even in the hands, which are very human-like, the finger bones are quite curved relative to ours. That indicates that it's very likely that this species was using their hands in locomotion much more than we do, climbing. Their feet, very much shaped like ours. Whenever I show the foot, I like to tell people that we have all the bones of the littler toes too. And in fact, toe experts can't always tell those little bones apart. And so I don't show a photo that has them all there because the foot experts say, you're just making it up when you do that. The toe bones, some of you probably have your third toe longer than your second toe and your fourth toe longer than your third. We don't know exactly those proportions in this species, but we know the rest of its foot was very much like human feet. A little bit shallower arch than our feet and a little bit longer lateral toes than ours. Its brain was around a third the size of today's people, 450 to 600 cubic centimeters. Its brain was overlapping in size with today's chimpanzees, with today's gorillas. It's smaller than many gorillas. So this is a species that combined characteristics found in today's humans in a very advanced way, and characteristics that are found in non-humans, living species that are not human. We called this Homo naledi, recognizing that it was something quite different from us. And as we continued to explore the cave system, we found other parts of the cave that also had bones. One of those parts is called the Lesedi chamber. It's UW102 in our numbering system. It's there in the center of the cave map. In that chamber, we found the remains of 3 individuals, including this partial skeleton of another individual, very similar in its anatomy to the first group that we'd found. This is the most complete individual today of Homo naledi that we've worked on. I was privileged to lead the description of this. And I will say that after all of this, I say I work with a lot of people and it's a collective project. We have at this stage described more fossils of human relatives than almost any other group of scientists in the world. There is an exception to that. I'll show you them in a little bit. Homo naledi lived around 250,000 years ago in this part of South Africa. Here are some of its skulls, and I'm going to put them on a map. Here in the southern part of Africa, you'll see our skulls, LES1, DH1, DH3, and there are other species of human relatives that lived at the same time in other parts of Africa. Some of those are members of our own species, Homo sapiens. We existed at this time, our species, and Homo naledi existed alongside of us. Our species was not alone 250,000 years ago when we first arose. Our species is very different from naledi in some important ways. Looking at their heads is the most obvious because of the different sizes of the brain. We, of course, think in human evolution that brain size is very important. Some of the things that we work on with Homo naledi lead us to think that brain size isn't always everything it's cracked up to be. but it certainly is a big difference between many of our relatives and ourselves and some of our slightly more distant relatives. Now I want to tell you about where human evolution was in 1925. And to do that, I want to put myself in the room, in a sense. This is me in Colorado, actually. I grew up in western Kansas, and I grew up in a town of 3,000 people. I had an excellent biology teacher, Gary Nelson, in high school. He was himself a laboratory scientist who went back to teaching. And so I got a pretty good background in biology, but I cannot say that I was thinking that I was going to be some kind of biologist. In the little town I grew up, that wasn't a career. And so I also had a really excellent English teacher when I was in school, and his name was Neil Melillo. And he was also the football coach, won the state championship a couple times. I guess he was better than Scopes. And he was certainly a great English teacher. And so when I went to university at Kansas State University, I set out to be an English professor. And so I was sort of majoring in English and got to take a course in anthropology. That's the sort of way that a lot of people get into anthropology. They haven't heard about it in high school before. We don't have anthropology courses in most high schools. I took a course at the university to learn more about it. I did pretty well in the course, and the professor, Mike Finnegan, who was a forensic anthropologist, he solved crimes. He identified the remains of Jesse James at one stage. He invited me to be a TA for his course. He said, you know, it's students like you that teach the labs in this course. I'd like you to come on and be a TA. Just take another course and then you can do it. We'll just pay you $300 a semester. And I did that. That was not my first paying job, but it paid more than any job I'd had before that, I think. And I'm really happy to say, I've got my wife Gretchen here today, I'm really happy to say that our daughter went to Kansas State recently and had the same TA-ship that I had. And so it's sort of a generational thing. From there, I became an anthropologist. That was what got me into it. And I have worked on fossil remains all over the world. Here I am working on one of the most famous Neanderthals ever found, the La Chapelle-Stance skull. But as I got into anthropology, I learned that it's sort of difficult to start working on fossils. People are kind of secretive about their fossils. They don't want to share them with you. And as a student, that was tough, because if I was going to work on something new and interesting, I needed to work on something new and interesting, and I needed to have the latest data, right? I needed to see things so that they could help shape my ideas, and I could be working on things that weren't behind the times. And that was just really tough. There was a time, the 1990s, when the Human Genome Project was new. And more information from human genetics was becoming important to the way that we think about humans and the way that we think about our evolution. And so I became a geneticist. I've done a lot of really fascinating work in genetics. My early work was focused on understanding how many people there were throughout our evolutionary history. How big were our populations in the past? And genetic data turned out to be a way that you could do that, because our genes are shared because of our genealogies, our relations. And those genealogies go back in time, and whether you're tracing the different lines of your family tree back to different people in tens, hundreds, thousands of generations in the past, or whether you're tracing them back to a smaller number of people, well, that's a function of how big the population was. If there weren't many people, your lines are going to become intertwined. And for people like me from a small town, probably for many of you, you know that small towns, sometimes those family trees intertwine. And you can trace your different cousins actually end up married to each other. They end up actually having an ancestor in common. And we can study that with genetics today. And of course, many of you probably, how many of you have done genetics like 23andMe or something like Ancestry.com, right? That's exactly the principle of this, is that you're tracing your genealogy back. Well, we can trace those genealogies back not just over tens of generations, we can trace them back thousands of generations. And we can study how people are connected. When you do that, you discover sometimes genes take unexpected paths. sometimes those genes have advantages that grow and accrue over time. How many of you all like to drink milk? Yeah, milk drinking is something that is, a lot of us like it. A lot of us, anybody have trouble with milk? Milk is a problem? Yeah, this is a normal variation. It's something that is absolutely normal. If I was giving this talk in South Africa, it'd be more, I'd say, probably 40% milk and 60% not. If I'm giving this talk in parts of Southeast Asia, I'd probably have maybe 10% milk likers and maybe 90% not. This is something that varies a lot, and it's because of our genetics. Lactase, the enzyme that helps us digest lactose, it digests sugar. Without that, the bacteria in your gut are happy to digest that sugar for you, and they create some gas and some unpleasant symptoms sometimes. And so if you don't make that enzyme as you continue to age, milk becomes harder and harder for you to grapple with. If you do make that enzyme, no problems. And whether you make that enzyme or not, as you age out of childhood, as you get older, is a function of the promoter of the gene lactase. And that promoter has mutations in human populations, all of which have arisen within the last 10,000 years in one place or another. So if you're drinking milk now, you like it as you're sort of adult, you share an ancestor, probably with almost everybody else here, who was the first to carry that mutation. And that person lived something like 8,000 years ago for most of you. And I studied the connections between humans and other species of early hominids, right? We even knew, before we had ancient genetics from ancient genomes, we knew that we could trace the ancestry back to ancient populations that no longer exist, populations like the Neanderthals. And I was, carrying forward my work on the fossils by understanding how their genetics might connect with ours. In 2010, I got pulled back into the field, into field work. And it was my friend Lee, actually, that turned me toward it. He had made some significant discoveries in South Africa, and his agenda was to open up the fossil record. He said, it's time to let everybody study these things. We're not going to keep them in a closet. We're going to promote them as much as we can. We're going to get them out so that everybody can see the fossils and work on them if they want. And his philosophy, which continues today, and I'm happy to repeat, is that if you're worried that someone else can look at your work and do it faster than you, then you ought to have them on your team. So let me look at 1925. Where were things? Where did they stand for human evolution at the time of the Scopes trial? I'm going to start with something biomolecular. This is Karl Landsteiner. He was a German researcher, and he discovered blood groups. 1900, he discovered the differences between A and B. Later, he discovered O. And those first differences, A, B, and O, right, which explained why you couldn't just transfuse blood from one person into another, that was the first biomolecular trait that was really characterized in humans and something that today we would call genetic. That discovery, 1900, corresponded coincidentally with the rediscovery of Mendel's laws of genetics. And blood groups are something where humans follow Mendel's laws. And this was clear to scientists during the 19-teens and 20s. This was something that promoted the movement of biologists toward thinking about human traits as being possibly Mendelian. It's because they could see that blood groups were. And so this was something that was known about human evolution at that time. The fossil record was actually fairly impressive when we look back at it. The Neanderthals had been well known since 1856, and in fact, the first Neanderthal skull had been discovered in the 1820s. The Neanderthals there were lots of them. It was quite clear by the 1920s that they were a distinct group, that they had been extinct for a long time, that they did not coexist for a very long time with humans, with modern people like us today, in the parts of Europe where we found them. So it seemed like there was really some sort of change where there had been Neanderthals once, and then there were modern humans later. There were some very large fossil samples of Neanderthals known. The largest at that time was from Krapina, Croatia, with hundreds of fossils. So the fossil record of Neanderthals, even in 1925, was very rich, and people knew quite a lot about them. They had some wrong ideas about the Neanderthals. And those wrong ideas sort of remind me of Neil's talk about the T-Rex. They had wrong ideas about dinosaurs. They had wrong ideas about Neanderthals as well. But the skeletal record was pretty rich. For humans and ancient hominins that are more distantly related to us, the record was much more sparse. In 1891, Eugene Dubois, a Dutch physician, had gotten himself stationed to the Dutch East Indies to try to find fossils of human relatives. He was convinced that they would be in the tropics and they would be in Asia. And he went there and he found them. There they were. He found these fossils. We call them today Homo erectus. He called them Pithecanthropus erectus, Pithecanthropus ape man. And today we accept that Homo erectus is a widespread species through a lot of our evolutionary history. The earliest fossils of it dating to around 2 million years old, the latest, according to many scientists, around 100,000 years old, so it had a long duration. And it lived in Asia as well as in Africa, and it lived there for a long time. In 1925, early in the year, was discovered this skull. This is from South Africa, and this skull is from a tufa cave called Taung. Taung was a giant tufa deposit. Tufa is a limestone that's formed by natural springs. If you've all been to Mammoth Hot Springs in Yellowstone, it's this sort of a formation. And they were mining it at that time for the pure lime. And that mining led to the discovery of some fossils that were in caves that were embedded within the tufa. And one of those fossils was this one, Taung. It was recognized by an anatomy professor named Raymond Dart. And he, when he saw it, saw immediately what it was. This is a child. It's got human-like teeth in many ways, as small canine teeth. It's not like any other living apes. The base of its skull seems to be configured in kind of a way like humans are, like it had an upright kind of posture. Its brain seems to be different from living ape brains in some really interesting ways. And Dart said, this is an ancient relative of humans. He called it Australopithecus africanus. This is still the first fossil of a human relative to suggest that there was an African origin of our lineage, that we had come from Africa. It was hugely important for that reason, and it's hugely important today. I was working not 3 feet from it last week in the lab where I work. And it is one of the most important fossils ever discovered because of that role in our origins. But it sketched the idea that there was something different in our evolutionary history than people had expected. Something that was an ape-like brain, this brain about 420 cubic centimeters, combined with more human-like teeth and other traits. That went against the other major fossil that was known in 1925. This fossil, Eoanthropus dawsoni, had been found starting in 1911 by a guy named Charles Dawson. And Dawson, when he was finding these fossils in a gravel pit, said, I've got this skull fragments, they're dark, they look like the other fossils there, I've got this jaw. Oh my goodness, it's a human ancestor. The gravel pit dated to the Pliocene, and so the scientists at the time in England all looked at it and said, well, you found a human ancestor, Dawn man, Eoanthropus. And they all stood by it. Arthur Keith, Smith Woodward, a lot of guys, they said, this is really real. It was fake. This is absolutely fake. Dawson had painted the things. Today, there's still some debate about whether he acted alone or whether he was working in conjunction with others. Some friends of mine at the Natural History Museum in London have concluded that the forger was a lone operative. There are file marks on one of the teeth that are really obvious if you look at them. Like, this was a fake that should have been picked up. It should have been noticed, and it wasn't. It was eventually recognized, but it did some damage for a while. It did some damage because with a human skull, actually a human skull, combined with an ape-like jaw, it set up an expectation among scientists that the first things that changed in our evolution were the brain. That's what set us on the path to humanity. And that led the same scientists who said that Piltdown is real, it led them to reject Australopithecus. They said, this can't be a human relative. Small brain is not how we started. Yeah, maybe it's got human-like teeth, but we know that we started with an ape-like jaw. So they absolutely had the backwards picture. I do not want to give the impression that in 1925 everybody thought this. There were lots of scientists, including American scientists, including William Gregory at the American Museum, who said, this is an ape's jaw and a human skull. This is not real, guys. They did not suspect at that time that it was a deliberate hoax, but they were quite open in saying, you found an ape and a human. They don't belong together. This is two different things. And so the scientific establishment, it's fair to say that there was published debate. It's fair to say that not everybody agreed, but certainly there was, it was widely celebrated that this was a real thing. Less widely celebrated was Hesperopithecus. Hesperopithecus, Harold Cook eye. This is more popularly known as Nebraska man. It's a fossil from Nebraska. You'll see it there, I believe, third from the left. This fossil, well, when Henry Fairfield Osborn at the American Museum, he was president of the American Museum, was sent this, he said, I think you found evidence of an anthropoid ape in distant past in America. This was Oligocene in age. In today's terms, that would be more than 35 million years old. He said, this is proof that humans had a start here in America. And he said, this is Dawn Man. Well, it is not. It was a fossil of a pig's tooth. And this in 1925 was accepted, but it was not long before it was overturned. This is a case where by 1927, additional excavations at the place in Nebraska where they had found the fossil turned up more complete jaws, and it was quite obvious that, oh wait, they have teeth that look like this too. And so by 1927, actually, the article that defined this was retracted. This is a case of science doing the right thing pretty quickly. It took until the late 1940s before Piltdown was actually under some sort of pressure to be retracted. If we look at the understanding of evolution at that time, here's Osborn's understanding. You see that the branches leading to gorillas, chimpanzees, orangutans are all there. The branches leading to the varied groups of humans are there. And some of the fossils that I just talked about are there. Now, what I want to point out on this tree is that the stems that go back from the four sort of skulls here at the left are all humans today. And this is a depiction of human races. And I want to point out that in Osborn's view, I'm going to show you next in Arthur Keith's view, that they all agreed that the races go way back. They thought that human races go back to the beginning of the Pleistocene. In their terms, that was old. They thought that was about 400 to 500,000 years ago. Today, we understand the beginning of the Pleistocene is close to 2.5 million years ago. So they thought races of humans were way back. They thought that the different fossils were extinct races, that they did not contribute very much, if at all, to today's people, although maybe they were close to the stem in some cases. The Arthur Keith's plot, this is from his 1915 book, you see that the long stems going to today's races, I've blown up that top of that image on the right so you can see the words. The races, Australian, European, African, he has them going back again to the beginning of the Pleistocene. So there's this viewpoint that there are transitional forms represented by fossils, that they are not direct ancestors of ours, but they're on somewhere close to the stem in some cases, and that today's races of people go way back in time, very far back. This is the only tree that was in the textbook used here in Dayton, and that was the exhibit in the Scopes trial. And I wanted to show it to you because when William Jennings Bryan was sort of lecturing to the court about this, he made a point of this graph. He said, what is this family tree? You've got 400,000 insects, right? What does that mean, right? How are they so important? And he said the mammals, There's 3,500 of them, and they're this tiny circle. He says, how dare you put humans in the tiny circle there with all those dangerous things, lions and tigers and bears? So he looked at this and he said, how can you put us into this tree? Of course, in biology, trees are our fundamental way of organizing things. I'm going to go from this, I'm going to show you today's point of view, right? What has changed? What do we know about human evolution today? This plaque is outside. Probably most of you have seen it at this stage. I wanted to use it as an example because the letters on this plaque are about an inch high. If you took our genetic code, every one of you's got 6 billion base pairs of DNA, more or less. It's closer to 6.3 billion base pairs. Women are a little more, men are a little less because X chromosomes versus one Y chromosome. You get the idea. But let's say 6 billion base pairs. If we were to print it out at the same size as this plaque, those 6 billion base pairs would form a square 2.5 kilometers across. It would cover mostly, if we were to write it on a monument, it would cover most of one side of the Great Wall of China. That's the information that we have from one person's genome today. It's an amazing amount of information. It's just huge. On the basis of genetic comparisons, by sampling genomes of different organisms, we can build a tree. The tree of life is multifarious and diverse. This is a tree of mammals. And as often is the case, when we have too many species to fit on a page, they make a circle tree, and the stems go back into the middle. So this is around 5,000 species of mammals depicted on this tree. Humans, you see there on the far right, have one little stem of this. The primates today, this is our order among the mammals, the primates. The primates consist of more than 500 species today, and we're again one of them. We understand their relationships in substantial detail. I'm going to talk about, this is the same tree, except I've simplified. I've taken only genera of primates, there's 80 of those, and I've listed the genera, and I've made it again a circle tree. And I'm going to use this as an illustration for a second. Our place in this is here near the bottom among the, among the hominoids. You'll see a gibbon swinging on one of those branches there. Homo is there at the lower right. Now let me talk about some traits that we understand today in evolutionary terms. How many guys have seen this before? All right, this is a color vision test. Is there anybody in the room that fails this? A couple of you, so several of you. That's not surprising. It's around 2% of men that fail this and a smaller fraction of women. The reason for that is that the genetics that are responsible for this are carried on the X chromosome. That means that men usually only have one copy of some of these color vision genes. They're called opsins. And women will generally have 2 copies of them. And so inheriting one copy that doesn't work quite right is unfortunate. Inheriting 2 is less likely. And so this is more common in men to be colorblind, as we call it, than it is in women. Color vision is caused by cells in your retina called cones. And the cone cells are called cones. They're on the back of your retina. They're called cones because they're different shaped than the rods. They actually are. When you take a cross-section of the retina, the rods are shaped like little sort of tubes, and the cones have this sort of cone shape. And they're arrayed on the back of your retina as sort of like the matrix of your computer screen is arrayed with different color dots. And so your perception of color depends on making those 3 different kinds of cones. The 3 different kinds of cones are different in their reception to different wavelengths of light because each of them makes a different opsin. Each of them makes a different protein receptor for light. And the opsins have different ranges of sensitivity. The red opsin is, we call it red because it's most sensitive to the longest wavelengths of light that are on the right that we perceive as red. The blue opsin is the one that is most receptive to the shorter wavelengths of light. They're on the right of this. We call them blue and violet. And the green is in between. Many of us see 3 colors. because our ancestors saw 3 colors. And we're related to a lot of primates that see 3 colors. All of the primates here that I've circled in the pink, that includes the apes and humans, the hominoids, and includes cercopithecoid monkeys, what we call old world monkeys. All of them see 3 colors. Many of the New World monkeys, the ones we call platyrrhine monkeys, also see 3 colors, but only females and only a fraction of them. Some lemurs also see 3 colors. All the other primates, they have 2 opsin genes. They see 2 colors. They're like dogs and cats. They see 2 colors like almost all the other mammals do. It's really primates that are odd in this way, and it's only particular kinds of primates, and within some branches of primates, it's only particular individuals. And again, within our branch, there's some individuals that can't. Why is this? In humans and in other cercopithecoid and hominoid primates, we see 3 colors because the long wavelength opsin was duplicated in our common ancestor. Instead of 1 gene, we have 2. A stretch of the X chromosome got copied again, randomly, as an unusual event in that ancestor, and that random event has been carried forward into all the descendants of that ancestor, including all of the old world monkeys and all of the hominoids. Only when one of those genes happens to break do you have somebody that arises that doesn't see the 3 colors. Occasionally, very rarely, someone is born who has another duplication and can see a little bit differently. They sometimes can sense 4 colors. So those random events are happening all the time. And in the ancestor of all these primates it happened. And that gene got copied, later changing its function to receive different wavelengths of light, along with part of another gene. You see that there's this block that's repeated. The L opsin gene, the long opsin, the M opsin gene, so that's red and green. And next to the M opsin gene is a gene called TEX28, and its full-length gene is there. And then between them is a cut-off version of that same gene. When this got repeated in that ancient ancestor, it copied one working version of the opsin and a partial broken version of the other gene. All of the primates that share this genetics share this broken gene. Broken genes are pretty common. This is the consequence of this, right? Why do these primates have this advantage? Because if you want to find food, if you want to find mates, if you want to avoid predators, it helps in the context where they live and where we live to be able to see 3 colors. Nocturnal primates have the opposite thing, right? If you are not active during the day and you're you want to be more sensitive to low levels of light, well, you've got 2 strategies. One is you can get giant eyes, which they do. This is Tarsiers' skull with gigantic eyes. You can reflect the light backwards. Like most mammals have a membrane in their retina called the tapetum lucidum. You guys, if you're driving on the highway at night, will see them shining back at you from the side of the road, and know to slow down, because if they're at eye level, that's a deer. So you see it shining, you know it's there, and that's reflecting your headlights back twice into their eye so that they really know that you're there, so that they can help raise your insurance rates. And many primates have this, but anthropoid primates, our branch and the old world and new world monkeys do not. You can change your coloration so that you're more recognizable to other members of your species. And that's happened convergently in very different groups of primates. The other thing you can do is throw out your cones altogether. Stop making the color opsins. Throw it all into rods. And so the short wavelength opsin has been deactivated in 2 different lineages of primates. So these are changes that happen all the time. I talked about blood groups at the beginning. The O blood group is another case where you're just not making the glycoprotein that displays on many people's red blood cells. Many people have an A, many people have a B. Those are active forms of that protein. If you're blood type O, how many of you guys are O? I am O as well. That means that you have 2 copies of a non-working gene. And you inherited that, most of you, from an ancestor that lived about 2.5 million years ago. Within the primates, we are the last of a once diverse group. That's what we understand today. The other primates that are close to us, chimpanzees, bonobos, gorillas, and orangutans, are themselves today diverse. Their diversity is greater than ours. There's species diversity in these different branches, 3 species of orangutans as we recognize them today, 2 of gorillas. There is depth of diversity in terms of ancestry of time. Their genetics are more diverse than ours are today. Humans were once diverse. We come from a diverse lineage, but that diversity has become extinct. We look at chromosomes. Chromosomes, most of us have 46 of them, 2 copies of 23, and those are numbered generally in order of size. Chromosome 1 is 1 because it's the longest, and chromosome 23 is, well, there's no 23. Chromosome 22 is the shortest. That has exceptions because under microscopes, you don't measure length all that accurately. Across our genome today, we can look at lots of different elements that are interesting and sometimes curious. One of those is called the ALU element. ALU elements are 300 base pairs long, and you have more than 1 million copies of them in your genome. This 300 base pair part of your genome does nothing except get copied again and again by master genes that put new copies in your genome. Odds are you have 1 or 2 that are new that nobody else had. You probably have a couple hundred that are variable and you have a copy and somebody else is different. There's more than 1 million of them across your genome, and many of them are all fixed in everybody today. They never vary. They mostly don't hurt. It's sort of like fleas. You sort of pick them up, and they mostly don't bother you, but once in a while they do. And one of them, around 35, 40 million years ago, caused our ancestors to change their vertebral development so that they didn't have a tail. So once in a while, they do something interesting. Mostly they do nothing interesting. There's different families of them. I'm showing you this sequence of them, the 300 base pairs, and there's about 6 different families that are illustrated here, and they differ from each other in sequence. So some of them share the different sequence variations, and each of them is like a barcode. We can recognize when this family was repeating in our ancestry by looking at the ALU elements and saying, oh, this is part of this family. Those families have been active at varied times in our evolutionary history. So in human origins, there's 3 families that have been active, and they've left a few hundred copies in our genomes. During the evolution of African apes, there's a couple of other families that were active. During the evolution of all of the monkeys and apes, there were some families that were active. And during the deep evolution of primates, there were families that were active. These are not found in other orders of mammals. So this is all stuff that's happened in our evolutionary history, and we share non-active copies with other primates depending on our relationship with them through common ancestors. This is a fluorescence micrograph of chromosomes, and it's circling here chromosome 2. And the reason why is that in this micrograph, the label that's being used to label things red under the fluorescence is a label that attaches to the telomeres, the ends of chromosomes. The telomeres are characterized by different sequence variations than most of the chromosomes, and so you can design a primer that will bind to them, and if you attach a fluorescent tag to that primer, you can make it glow, and that's what they've done here. They've put a red glow onto the telomeres. But this chromosome has the glow in the middle. This is chromosome 2 in humans, and chromosome 2 in humans corresponds to 2 different chromosomes in our close relatives. Chimpanzees have 2 different chromosomes. One of them corresponds to one branch of our chromosome 2, the other one corresponds to the other branch. Gorillas, same story. We've had a fusion of our chromosome 2, and that fusion in our ancestry put the telomeres in the middle. And they're still there today. The ends of the chromosomes are stuck in the middle. because of this random event that once happened. Going a little bit further out, we know that our relatives are interbreeding a little bit today. We talk about species. We've changed our understanding of species quite a bit in the last 100 years. Species interbreed sometimes. Bonobos and chimpanzees separated about 2 million years ago. They interbred once in a while during that ancestry. We can detect that with genetics. Gorillas interbreed once in a while between their species and have input from an ancient gorilla population that no longer exists, an extinct population. Humans have input from a lot of extinct populations. One of those is the Neanderthals. I remind you that Neanderthals, we had a lot of evidence of them. We still have a lot of evidence of them today. And today we have their whole genomes. This is Vindia Cave, beautiful place where I've been a couple of times. These 3 bones from Vindia Cave each have given rise to whole genomes of Neanderthals. We have Neanderthal mitochondrial genomes that show us a tree of relationships among them. We know a lot about the genetics of Neanderthals today. From deeper time in the Neanderthals, from the Atapuerca mountain in Spain, deep in another chamber is a group of people who have found more fossils and described them than our group has. They work on fossils in a place called the Cima de los Huesos. And those fossils are fossils of very early Neanderthals, Neanderthals that lived around 450,000 years ago. There are more of them in that deep chamber than there are of any other fossil site in the world for human relatives. And one hand axe. They call it Excalibur. We've got so much evidence of fossils. I got to tell you, right? A lot of times people make a big deal out of Lucy. We've got evidence of fossils from many different parts of the world, whole skeletons, partial skeletons, sometimes a tooth, and sometimes bone fragments within sediment that are so small that you have to use a magnifying glass to see the bone fragments, but these bone fragments still contain DNA. This section of sediment is from Denisova Cave in Siberia. Denisova Cave is famous for this finger bone, which generated the whole genome of an individual that lived around 80,000 years ago in this case, and that individual was not a Neanderthal, they were a different group, a different population. Denisova is a really unique place. You walk into it, it's like a walk-in refrigerator. It's cold and it preserves the DNA well. It preserved the DNA inside of this little bit of bone from an individual that had a Neanderthal mother and a Denisovan father. Hybridization is part of our ancestry. It's something that's become very clear in the last 20 years. The Denisovans, we have bones of them that we've identified biochemically throughout parts of Asia, and this skull, which just this last month was identified biochemically as Denisovan. So we now can connect the fossil record of East Asia, individuals that lived at the time of Neanderthals in the West, to genetics. Our tree is a tangled one. It's complicated. It has lots of twists and turns in it. I want to sort of close by talking about the breadth of the record, right? The kind of evidence that we have. I'll take you back to the site where I come from. This site is Malapa. It's a site where I've been privileged to work for a while. I'll walk you up to the Malapa site. It's a little cave. It's in the same region of South Africa with the Rising Star Cave. It's set up for tourism. That's why we have a boardwalk, and you're going to see us walk out over the site. And this cave has generated 2 skeletons of a species that we named Australopithecus sediba. That species is one of the closest relatives of Homo, our genus, out of all of the different kinds of early hominins called Australopithecus. And it's one of the most completely known. I'll walk you up over the site. You'll see down into the site here. Here's where the fossils come from. It's a very unimpressive little cave. You don't have to endanger your life to go into it. That's all it is. But in it were these skeletons. Here's a young male individual named Karabo. We know he's male from protein sequencing, actually. And the rest of Karabo, my friend and our preparator, Zandile Ndaba, has been bringing out of this rock for the last 3 years, completing this skeleton. We have now one of the most complete skeletons of any early hominin. We know a lot about its anatomy, but one of the most important things we know about it is its hands. They're human hands. I don't have to tell you, right? This is a very non-human looking kind of creature. In some ways, they stand and walk upright in the way that we do. They have human proportioned hands. The distribution of bone inside of their fingers is distributed like human hands. They're using their hands in the way that we do. Right? These are the connections that today we work to study. We find the ways that ancient hominins behaved. We connect it to the anatomy, the hard evidence, the stone. We increasingly can connect that with biomolecules, which are a volume of data about relationships and connections and function. We're talking today about digestion of Neanderthals. We're talking about the way that their genes inside of you, most of you have about 2% of their DNA, are still functioning. They're there within us. And so when we look at the bones, one part of understanding them is understanding their lives, but a more important part is understanding how all of this evidence contributes to our bigger picture of where we came from and how we work. And I can tell you that today the forefront of human biology is right where we are, understanding why humans get fat, why we have trouble with today's diets, why our microbes might be going wrong, what's happening with diseases. It's happening now in the fossil and archaeological record as fast as it's happening anywhere. We can trace the ancient diseases. We can show what our susceptibilities were. We can see what the digestion of ancient hominins was. We can study the composition of their bones to study how they formed. So it's an amazing world of science. I'm really happy to give you guys a picture of it. I really regret, on behalf of our organizers, for going longer than I should have. But I want to thank you all for your attention. I want to thank especially Tom for his efforts in organizing this, and all of you here in Rea County for this wonderful event. Thank you all.
We've got time for 2 questions. Okay, we got one right there.
You called the questions. Yeah.
I was wondering, how do you know where to look for fossils in the whole Earth? Where do you look? And how do you know that when the pieces fit together,
Yep.
it's the same organism as opposed to like Piltdown Man or maybe
Yep.
two?
That's a great question. In fact, today, I'll take the second part of that first. We are today so skeptical about whether pieces fit together. I think that's one of the biggest lessons in the history of science, right? You find 2 things next to each other. There's a coincidence there. You found them next to each other. You have to ask, is this a coincidence that has some sort of cause behind it or not? And we today, I spent most of the last month studying this little baby skeleton. And it's a baby of Homo naledi. It's the most complete early hominin-looking baby that we've ever discovered. And I will tell you that every bone of that, we found every one of those bones within a square this size. And nonetheless, every one of them I look at and say, can I be sure that this belongs to this kid? Could this possibly be somebody else? And I found that skeleton in the Lassetti chamber, the neoskeleton. And the left and right femora are a little different from each other. They're the same size, more or less, but they have some little tweaks. And the folks that I work with that study lots of leg bones said, we're not sure those belong to the same individual. So we published, we're not sure they belong to the same individual. It's unlikely that they don't. And in fact, today, this has led to a huge discovery on our part because we've found asymmetries in other parts of the skeleton that actually tell us that this individual had some kind of syndrome that affected their skull, their vertebrae, and their legs. And so it's giving us a clue. But I will say that we approach it that way every time. The question of how do we know where to look? The answer is you look everywhere. I really want to emphasize that most of the discoveries in this field have come from making a road and a road cut, right? This is true of archaeology in North America. That's why we do archaeological survey before the highway starts building, right? Most of the discoveries that we have are made by people that are not experts, that are not looking for things. I do work in a part of the world where we have caves that people are in a lot, but they're not necessarily looking for fossils, and it profits us to go and look and to sink some trenches and see if we can find some evidence of habitation. But the answer to your question is they are everywhere because ancient people and our relatives lived across the face of the planet. So we find them almost everywhere that they would have lived. Yeah.
So, two of the expert witnesses that the defense wanted to call in the Scopes trial invoked Piltdown Man as evidence.
Yeah.
Of this. And can you tell us just how discredited was the Piltdown Man discovery, or whatever you want to call it, so that it wouldn't have actually discredited the testimony of those witnesses?
That's a great way of approaching the question, very legal. It's the fruit of the poison tree, right? It absolutely is. Man, when I look back at Piltdown and see the people that I read a lot of their work, people like Franz Weidenreich, who later became well known for discovering and describing Peking Man. When I look at these folks and how they write about Piltdown with complete dismissive language, they really say, if you're going to put together a chimera, you might as well put on the goat's head and the fish's tail, right? They really, really went after it at the time. And so there were real scientific skeptics. I will say that the folks who were promoting Piltdown, Arthur Keith in particular, were equally harsh on the Tong skull, right? They said, I don't believe this is anything but an ape. This is not connected with humans. Yeah, it's interesting, sure, of course interesting discoveries could happen in Africa, but we all know that the real ancestry of humans came from England, right? Clearly, right? And there was so much bias in it. I will say that the scientific story of this is one where radiocarbon dating fixes everything, right? Oh, they dated it, and oh, surprise, it's a fake. There are file marks on the teeth. This is one where if the fossils had been examined in the way that we today insist on examination and sharing of data and displaying your data, it's like the old math thing, you show your work, right? That's how scientists work today, you show your work. When I have colleagues who will not show their work, who say, oh, you can't study my fossil, I'm not afraid to say, you're not doing science. That is not science. If you're not making that available for other people to study, that is not science. Because it's not. So I can give them some, forgive them a little bit. In the era, it was hard to, provide access to everybody when you had to take a steamship to do it. But the reality is that, people took a lot of things on expert authority. And today we very much don't, right? Today we very much say, yeah, if this is real, you'll show me the data.
John, thank you so much.
Thank you all. Thank you.
That concludes this morning's session. We'll be back at 1 o'clock and take a look at Christian reaction to evolution. So enjoy. Let's